contain the microsporangia, like located at every corner, like all the four corners like that. And ultimately, we can say that what are the features of the anther? The anther is bilobe. First of all, it has two lobes. It is surrounded by two theca, so it is dithecous, and it is tetrasporangiate, meaning it has four sporangiate structures present. Now the microsporangia, they develop and they become the pollen sac. So inside the tetragonal structure, there is the presence of microsporangia. Now these microsporangia only, they're going to develop and become the pollen sac. And inside the pollen sac, there will be thousands and thousands of pollen grains. And these pollen grains, they are the male gametes of the plant. Just like how sperm is the male gamete of the humans, right? Same way, the pollen grains are the male gametes in the case of plants. Now, when we talk about the structure of microsporangium, so if you take a microsporangium and you cut it into half, you will see a circular outline. And these are surrounded by the four wall layers. Epidermis, which is the outermost layer, if you can see over here, outermost layer is the epidermis. Inner to the epidermis is the endothecium. Then inner to the endothecium, we have several middle layers, 2 to 3 middle layers. And the innermost layer is the tapetum. So four layers are going to make up the microsporangium wall. Now see, each of these layers, they have their own particular function to perform. For example, epidermis, endothecium, middle layers. The first three layers from the outside, they are responsible for protection, first of all. And apart from that, they help in the dehiscence of anther to release pollen. Now see, once the anther has matured and enough amount of pollen grains have been made. So then what happens? The anther, it bursts. Why? So that it can release the pollen grains into the air, into the atmosphere. So this bursting of the anther, we call it as the dehiscence of anther. And this phenomena is actually, you can say, happening because of the first three layers from the outside. Now the innermost layer is the tapetum, and this is solely responsible for providing nourishment to the developing pollen grains. Right? It provides nourishment. Now the cells of the tapetum, they possess a very dense cytoplasm and they have more than one nucleus present in them. They are usually binucleate. They have two nuclei present. Now furthermore, present at the center of each microsporangium. Imagine, we started from anther. Inside the anther, we had microsporangia. Now, inside the microsporangia, there is the homogeneous cells. Homogeneous means same kind of cells called as the sporogenous tissue. And these tissues, they undergo the process of meiosis, which is reductional division, and they give rise to the pollen grains. This is how the pollen grains are made. Okay, any doubts? Sure.
Yes, ma'am.
Okay, you know down till here. And if you have any doubts, feel free to ask. Okay, because this chapter you have to learn a lot of technicalities, so everything must be clear. Done. Sit here.
Ma'am, I can't hear you.
Shall I slow down?
Yes, ma'am.
Okay.
Done, ma'am.
Done, Justin?
No, ma'am.
Okay, okay.
Done, ma'am. Ma'am. Done, ma'am.
Okay, now we're going to see how the process of microsporogenesis happens. Now, microsporogenesis, see, the formation of microspore will be called as microsporogenesis. So now we're going to see how this entire process actually takes place, what goes behind it. So when we talk about microsporogenesis, it is the process in which the pollen grains are formed from the pollen mother cell. The other name for pollen grain is microspore. That is why we are calling it as microsporogenesis. So here the pollen grains and how they are formed will be called as microsporogenesis, and these pollen grain or the microspore, they are formed from the pollen mother cell through the process of meiosis, because obviously you tell me why is meiosis the case? Switch on your video. Why is meiosis being used for the formation of pollen grains? Hmm?
Because if you use mitosis, it will be equational division.
Hmm. Very good. Because for the formation of gametes, right? Reductional division is important because the gametes that have to be formed will be N, right? Haploid. So now when the anther develops, the cells of the sporogenous tissue, they start undergoing the process of meiosis to form a microspore tetrad. So it forms 4 cells of microspore. So what happens? The sporogenous tissue, first of all, they divide by mitosis and give rise to microspore mother cell. Then the microspore mother cell, they divide by meiosis and forms microspore tetrad. And this microspore tetrad develops into pollen grain. But before this, you have to see here that the microspore tetrad, it is like a cluster of 4 cells. This one. But before directly forming the 4 cell stage, there is the formation of 2 cell stage, 1 cell stage. So from the 1 cell stage, like the microspore mother cell, it will develop into microspore dyad, which will have 2 cells. And then into a tetrad, so 1, 2 and 4. Then 4 cells will be formed. So inside each microsporangium, there are several thousands of microspores which are released with the dehiscence of anther. So several thousands and thousands of pollen grains are developed inside the microsporangia, and once the anther matures, then it will burst, ultimately releasing the pollen grains into the air. Now, when we talk about the pollen grain, so it is the male gamete and it is usually spherical in shape. The diameter of a pollen grain is 25 to 50 micrometers and it has 2 layers, the outer layer and the inner layer. The outer layer of the pollen grain is called as the exine, whereas the inner layer of the pollen grain is called as the intine. Now, both of these layers are very important, especially outer layer. Outer layer, it is hard, it's very hard, and it is made up of a compound which is called as sporopollenin. This is very important. Okay, so the exine, it is made up of sporopollenin and it is a very resistant, very hard substance that is present in the atmosphere. Now, why is it so hard and resistant? Because it can withstand high temperature and strong alkalis. Like there is no base, there is no acid that will be acting on the sporopollenin and degrade it. Nothing can degrade it. So no enzyme, no acid, no alkali, nothing can degrade sporopollenin. Now the exine of the pollen grain, it has a prominent aperture. So there is like a place where the sporopollenin is absent, and that area is called as the germ pore. So it's like a whole entire area Sporopollenin is present, and at one place the sporopollenin is absent, and we call that area as germ pores. Now see, because of the presence of pollen grains only, or because of the presence of the sporopollenin in the pollen grain, the pollen grains are very well preserved. Why? Because of the sporopollenin, it will not get degraded. Then the innermost layer is the intine, and it is continuous. It has no gaps, and it is made up of cellulose and pectin, two things. The cytoplasm of the pollen grain, it is surrounded by the plasma membrane. It also has a plasma membrane, and see, once the pollen grain is mature, it has two cells in it. This bigger cell you see, this one, this whole entire thing. This bigger one is called as the vegetative cell or the tube cell, whereas the smaller one, which is called as the generative cell, it floats in the cytoplasm of the vegetative cell. So two cell stage. Okay, these are two cells in one pollen grain. So talking about the vegetative cell, it is the bigger and has abundant of food reserve. It has a large, irregularly shaped nucleus. Whereas when we talk about the generative cell, so it is the smaller one and it floats in the cytoplasm of the vegetative cell. Talking about the shape, so it is spindle shape and it has a dense cytoplasm and the nucleus. Clear? Now, this is one of the most important lines, that in 60% of the total angiosperms, pollen grains, they are shed at two cell stage. Whenever they are at this stage, generative cell and vegetative cell, they will be shed, like released. But in the remaining 40%, what happens is that the generative cell, the smaller one, it divides by mitosis and give rise to two male gametes before shedding. So now it's like this situation. Okay, so one bigger one, 2 and 3, like this. This is a 3-cell stage at which in 40% of the rest of the angiosperm, the pollen grains are released. Clear? Any doubts? Okay, now you can note it down.
One man.